Okay, this is our first attempt to discuss a paper. Hopefully this will work out well. I always get nervous when I discuss papers because I honestly consider myself still like a student of medical veterinary entomology. I'm always still learning. And every time we read a paper, I'm trying to push my knowledge. So I don't have all the answers. So if you guys participate, that will be great. And we can all learn from each other. So this was the paper. Mosquito saliva enhances virus infection through sialokinin-dependent vascular leakage by Daniela Lefteri et al. Okay, this is just recently published 2022 in PNAS. Okay, so first off, let me just give some, since this is the first paper, we're discussing some thoughts about papers that you should keep in mind. I don't know what the exact percentage, but there's a stereotypical heuristic in science that essentially like 9 out of 10 papers that you see published are probably actually like trash. So don't ever read a paper and assume because it has a good author or because it's in a nice publication that it's true. So you should always be skeptical. When I read papers, I kind of try to find things that are important for my research, and I try to I don't necessarily believe that it's necessarily true, but I sort of take these new ideas into my mind, and then if I encounter multiple papers that are sort of repeating that idea, then it sort of to me becomes more true. So just keep that in mind. Don't take this as true. Okay. With that thought, it's always helpful to think about papers. I always try to think about what are the questions that they are asking. So let's try to brainstorm and enumerate some of the questions that you think this paper is asking. Any thoughts? So you're already too specific. What's the bigger question above that? That's right, but yeah. Does saliva influence pathogen transmission? That was like the main thing that I got. And you're right, like later on, after they answer that question, they try to figure out, okay, what is it in the saliva? But basic to start with, it's just essentially like, does saliva influence pathogen transmission? This is an interesting question. Why would this evolve? Like, how is this happening? How is it possible that saliva could influence pathogen transmission? And anything in biology, it means there's some evolutionary context that sort of like draws that out. How would this evolve? And why? Okay, wait a minute. You said virus helps. Say that again. So this is a very interesting thought. Okay, so one perspective is perhaps the virus helps feeding. This would be the idea that this evolves from the perspective like the fitness advantage would be in the case of the mosquito. So this would be like helping mosquito fitness. Although I think that's unlikely. I think it's more, what are you going to say? Like help the mosquito? There are some ideas that maybe you get, if you're a mosquito, you get some pathogen that like you somehow like, maybe it would tweak your feeding behavior. Maybe you would be more likely to get a blood meal. But I think it's probably less likely that that's true. I think the mosquito probably does not want to be infected with a virus. The virus is going to be taking a toll on the mosquito in most cases. Like it's going to be infecting itself. It's going to be stealing energy to reproduce. So I think, while it could be true what you said, I think it's less likely that this thing evolves from the perspective of the mosquito, like the mosquito having beneficial fitness. What's the other perspective under which this could evolve? You mean if there's a sick host, they would be easier for the mosquito to feed on? That's an interesting question. Yeah. Like they're going to be laying there essentially like sick in their bed, and they yes. Yeah. So I didn't think of that. That's a great answer. If the sickness makes the animal sick and it's not moving around as much, it might be more easy for more mosquitoes of that similar species to get blood meals. And then that could, in a way, help the fitness of the mosquito. That's an interesting thought. That's what I was looking for. Yes, that's exactly what I was looking for. So what I was thinking of, like, there's two ways to think about this. One would be perhaps this comes about from the perspective of the mosquito, like the mosquito gets a fitness advantage, or perhaps this is just the virus figuring out a way because it has to replicate itself. And so it's figuring out like it has to co-evolve with the mosquito. So it's just sort of figuring out, okay, like what are the tools that the mosquito's spitting in? How can I, any way that a virus is going to be able to use that to its own advantage, it's going to create situations where that virus replicates more. So that's more what I was thinking exactly. It's probably that the virus evolved this relationship. So it's not necessarily that like the spit evolves in the mosquito to promote virus pathogen transmission, unless your idea is correct. That could be correct. It's probably more true that this spit is already there and the virus is figuring out a way to take advantage of it, if that makes sense. Good. This is great. Okay. There is another question, sort of like broad question that this paper, they kind of talk about in the introduction. Did you guys catch it? Any other reasons why you might do this? What do you mean? Like that's a really good one. They do address that later. Essentially, like why do anopheles not spread famous like horrible viruses? I mean, there's some that they spread, like they talk about the Ong Yang Yang virus. No, that's definitely hard. So that is a question. Is like, why do certain mosquitoes, that's what we talked about yesterday, the vector competence, or last time, why do certain mosquitoes have a high vector competence for some pathogen but not others? And it could be in the spit. So why do anopheles not spread versus Aedes in this case spread many like flaviviruses and arboviruses? Yes, that was the one that I had. Is that's exactly right. So can you explain that? So there is a specificity in cell types. That's true. But I think that main point is more about like, and this is the case for most arboviruses, where if you say if 100 humans get some arbovirus, most of the time it's only like a small fraction of them that have horrible disease pathology. And so it's like, what is it about those humans that makes them particularly susceptible to these diseases? And is there some kind of a component in the mosquito that they are extra susceptible to, or something like that? And so that could be, it could be, I think they're hypothesizing that it could be a situation where something in the saliva that the mosquito is spitting in is doing something to that human's biology to contribute to the pathogen transmission. So I'm going to put host susceptibility in terms of like why some humans are worse off than others. if they get an arbovirus, etc. These are definitely like, this is good. This you guys are showing, demonstrating you have a good understanding of this. These are definitely kind of like the main points that I got. Okay, so I'm debating what to go into next. Okay, so then after I sort of like map out the main reasons like why you're doing this study. Okay, if you notice, they go through it kind of like hypothesis by hypothesis, experiment by experiment. So the first experiment, we'll call this experiment 1, is test if mosquito saliva can enhance virus infection. Okay, and I'm going to grab a figure, which is figure 1. Okay, and I want you guys to kind of like walk me through. I'm just going to grab ABC. So also what you're going to notice when you read these papers, some of these papers are like ridiculously long, and papers are getting longer and longer and longer, especially in the top journals with more and more and more supplemental data. So even me, like if I have an important paper, I'll try to read it cover to cover, and I will read it multiple times if it's an important paper. If it's me kind of like trying to branch out, I don't necessarily look at every figure, but there's kind of like money shot figures that like prove their point. And I try to go through and understand those. So with respect to this experiment 1, here's some of the key data. Let's set up what they're doing here to help facilitate the discussion. They are step 1, they are collecting saliva. They are step 2, oh, let me move this up. Step 2, they are co-injecting alpha virus or Zika, that's Zika virus, with the spit or without the spit. Okay. And then step 3, they are measuring with qRT-PCR. So let me just make sure everybody's on the same page about these things. Step 1, that's easy. Okay. Step 2, this is a good question. Why are they using these 2 viruses, alpha virus and Zika virus? Maybe this is a broad question, but it's actually an important scientific point. Why are they using 2 viruses? Oh, is that why? That's something I did not catch, if that's true. Is alpha virus? Okay, so here's where okay, so now I'm going to look bad. Let's Google alpha virus. I thought alpha virus was a arbovirus. Yeah, that's what I thought too. But maybe no. But you said it's not spread by insects? I don't actually know. Did they say that? Oh no, it is. So it says terrestrial alpha virus infections are spread by insect vectors. Okay, so that's why that caught me off guard, because I did think that both of these are spread by insects. But more broadly, so yes, you're correct. Both of these are arboviruses, like so that's what they want to study. But why do 2? This is like, think about this. This is actually a very important sort of scientific method type question. Why are they studying 2? Yes, that's actually like a really important point that's worth pointing out. I have a term for this. I call this convergence. Convergence is when like if you have a phenotype and you test it in multiple systems and you see the same thing in multiple systems, that's way more evidence that that phenotype is real and exists than if you just demonstrate one thing interacting with one other thing. That could be just a fluke. That could be some random experimental thing. So I'm always evaluating papers based on like a paper is very good if they have this convergence thing where they can see a phenotype in multiple systems. That suggests it's real. Something is really happening. So that's why they have 2. The other reason they probably have 2 is because like when they were first starting this off, like maybe they actually tested with 10. Who knows? Like they probably tested this with a few different viruses. And so they're kind of like looking for a phenotype and they're finding the systems where the phenotype is observable and measurable. It's probably actually what they're doing. And then they're reporting that they saw this phenotype in 2 systems. So you don't see what they also tested but got negative data.
Trying an alphavirus, virus, there are other alphavirus like
Yeah. Yeah.
But those are kind of hard to have, or
Oh, so alphavirus is a model, kind of like, yes, for other viruses. Yeah.
Why?
Mm-hmm.
I don't know.
Yeah. Yeah. So that's another good point, is that these are the alpha virus is a model. The Zika virus is nice in a sense of Zika, you can actually study under BSL-2. So you don't need a ridiculous like BSL-3 or 4 facility to study these. So that's another good point, is in some sense they're model viruses. My point is just like, don't ever just study one thing. Always try to find that same phenotype in multiple examples. And then you have much more broader evidence. Okay, so step 3, just to make sure everybody's on the same page. Does there anybody who, well, I phrase it like that. Does everybody know what QRT-PCR is and how you measure it and why they would use that technique? Like, what is it measuring? QRT-PCR. First of all, what's it stand for? Yes. Quantitative. So that means you can quantify it, you get a number. Reverse transcriptase. What is the function of reverse transcriptase? Yes, it turns RNA into DNA. So it's a very odd enzyme. It's from viruses. And it's technically, it's a, let's see if I can get this right. It is a RNA-dependent DNA polymerase. So that's how it can turn RNA into DNA. And it doesn't quote unquote turn it. What it does is it will bind to an RNA sequence. It'll then, what would you say? It would write a matching DNA sequence to match that single-stranded RNA molecule. Then from that, so the first part of QRT-PCR is you do the reverse transcriptase. That's part 1. Part 2 then is you do a PCR, which is amplifying that DNA product. And that's what you quantify. So on most of these graphs, on most of these charts on the y-axis, you are seeing essentially like copy numbers of RNA from these viruses. So it's essentially, if a virus gets made, it'll replicate its genome, that RNA will appear, and however many copies there are, that's a good estimate for how many viruses there are. So the y-axis is essentially like number of viruses. You measure that by QRT-PCR. Okay, so let's walk through these data. Let's first look at A. When I look at figures, I always like orient myself first around the axes, and I always try to figure out like, what are they comparing? Okay, so in this one, they're comparing skin, inoculation site, and in this one, they're comparing blood. Okay, so why are they measuring 2 different, I guess, what would you call this? Samples? Tissues, yeah. Why are they measuring 2 different tissues? Yeah. Yes. Yes, that's very well said. So the skin is essentially like the bite site, and the word that was said was local. That's like, you might be able to promote enhanced virus replication at the local site, but that doesn't necessarily mean you're going to get a better systemic infection throughout the body. Okay, so that's why they measure the skin. And then they're also measuring the blood, because that is like what the word was viremia. That's actually like, if you're getting sick, you have a systemic infection that's circulating through your blood. And so you'll be able to measure the virus in the blood. And it's totally reasonable that you might see an effect in the skin, but maybe not in the blood. There might be some organismal barrier or something where you would see one, but not the other. And what they hope for, which they observe, is that they do see an effect in both. Okay, so this is the alpha virus. Hopefully I'm getting this right. And this is the essentially same data, same type of experiment for the Zika virus. Okay. So what would you, what's the, okay, the other thing I do when I'm looking at figures is what's the negative control? So like in this first part of A, what's the negative control? Yes, SFV alone. So this is alpha virus by itself. Hopefully this is not too obvious. And you're comparing everything else to that. Obviously, you can tell from the statistical comparisons. So anytime you add saliva, so they've milked the saliva from the mosquitoes, which is really interesting. And then they're injecting either 1 mosquito's worth or 5 mosquitoes worth or 25 mosquitoes worth. It's always increasing. Ditto with the blood. Okay, so this is actually a really easy figure to interpret as long as you just take it component by component and understand the axes. Everybody kind of understand this? Okay. Let's see. Ditto for the Zika, like it's the same thing. Let me see. Oh, this was really funny. This was actually really funny. What is this? What's happening here in C? Yeah, so this is a so what would you call that? Why would you simulate probing? And how are they simulating probing? They're stabbing people with like a tiny, they said it's like a micro needle, and they're like stabbing the people 10 times. Why would they do that? And what is this? What's the word for this in science? Number C. Very broad word. This is another big like negative control. So oftentimes, again, like maybe I don't know how comfortable people are reading papers, but a lot of times that's what I mean by like the money shot. A lot of times in figures, it's kind of like a little part of it that's like the part that's telling you the result, and then 3/4 of the rest of the figure is like negative controls, which is very important. But yeah, anyway. Okay, so they're stabbing them as a negative control. Very interesting. Let's grab the next part of this. Oops. I want this. Let's grab D. Okay, what's this? What's happening here? Survival rates of what? Of what? Of mice. That's always important. Like learn, note what they're testing it in. Okay, and it's a survival rate. So essentially, like, what does a drop in this bar mean? It's a death event. So you have some population of mice. This is very common in mouse research. You see this in fruit fly research. This is essentially like a survival curve. Any tick down is a death of an individual from the population. And you're essentially just comparing like how fast they die and how many die. Okay, and the key take home is that if you have the Aedes saliva or the Aedes bite, they're dying faster and more of them are dying as opposed to just the virus alone. So this is kind of like, I would say this is probably the main piece, one of the main pieces of this paper, because what is this essentially like prove? Or maybe not prove, but like what is this strong evidence for? Yeah, the saliva enhances what you maybe, yeah, mortality. That would be a good way to say it, mortality or pathogenesis, mortality or pathogenesis. So this is like, that's kind of like an important figure. Okay. Okay, this is the next thing. The next experiment that they do, if you read the paper closely, you actually don't see figures for the next experiment because the next experiment like essentially failed. And it was to test if the enhancing effect is organismal, cellular, or the virus. I'm trying to think, what do I mean by this? Essentially, oh, I remember now. Okay, they tested. Where did they test this next? Alec, you should have noted this. What did they test next? They tried to replicate this sort of like enhanced viremia data. Yeah, they tried it They tried it in vivo in cell culture. Yeah, so this was interesting. They tried essentially kind of like the same experiment, like you have a cell culture and then they're like inoculating them with some virus somehow, and they're like adding in spit. And they did not. It was actually reverse. I think it like protected them, which was bizarre. So the saliva, I didn't look at the data. It was all in the supplemental. Why do you think they shoved it in the supplemental? Exactly. So again, like that's another reason. And I'm not saying that I actually think this paper is like pretty good, but yeah, that's why they shoved it in the supplemental. It's because when they tested it in cell culture, the saliva actually protected. So the effect did not reconstitute. So the reason that they would do this is because this lab probably has a lot of experience with cell culture. And so they probably got this great phenotype in the mouse, and then they're like, let's just see if we can also replicate the phenotype in cell culture. And then that would sort of like, if they got it, that would open up a whole different realm of sort of experimentation that they could do. So they probably just quickly tried it and it didn't work. What was your question or point? They were using mosquito cells. So I looked at the what? Wait, no. Because they're probably using human cells at this point. Oh, I remember, I wrote it down. They first tried fibroblasts. So they're using human cells. Fibroblasts, which are what type of cells? Those are skin cells. So fibroblasts are skin cells. And then they also tested macrophages, which are, now say it. What? That's an immune cell. And that's actually key to their hypothesis, which we'll talk about a little bit later. But they tested these with 2 different cell types, and the effect didn't happen. And then they also, they didn't give up. They actually tried like dissections of like chunks of skin. So they pulled out chunks of skin and tried to replicate it, and it failed. So let's see, what's the take home from this? The fact that they got this great phenotype in this figure 1, and then they tried to replicate it in cell culture and it didn't reconstitute. What would be your take home? Yeah, so that was their sort of like pivot. Is like, we're not seeing it everywhere, so it must be like an organismal. And I think it turned out well for them. Like it's an organismal phenotype. Can you explain that? Like how would there be a phenotype that would not exist in a cell culture, but would exist in an organism? Yeah, that's a good way to say it. Like a multicellular organism is vastly complex. And it's actually in science, it's very, very common to see totally different phenotypes for a cell culture experiment versus an organismal phenotype. That's common. And so they're sort of like how they like navigate this nuance is, in your opinion, what do you think is more valuable, the organismal data or the cell culture data? The organism data is more valuable because that's what's actually happening. And so you hope for a situation where the organismal data looks like the cell culture, but you don't always get that. So they sort of just pivot and they say, okay, it must be like an organismal, I spelled it wrong. It must be an organismal phenotype. Okay. Okay. So then the next step, let's call this experiment 3. What would you do at this point? So you know you have this phenotype, it's probably an organismal phenotype. What would you do? Or what did they do? What's the next step? But why are they doing that? But why are they doing that? There's a broader, like let's test this. Yes. Yeah, the next, so that's the next experiment is what is causing the effect. Okay, and this is sort of like high level science versus low level science. Is you might be able to find a phenotype, but now show me like what's doing it. That's high level science. So that's what they want to figure out. What's causing this effect? And so y'all listed like a battery of tests that they started doing. But even before they do that, like they first sort of developed like a list of possible like explanations for what could be causing this effect. in the saliva. And what was on their list? People who were saying what they did. Yeah, so heat, but that's actually the heat treatment is testing what hypothesis? Right, so you're testing the hypothesis, is it a protein? If it's a protein, it should be able to be inactivated by like a heat treatment. Okay, what are the other hypotheses that they detect or test? Yes. So it's gonna fuck with me. Okay, so yes, like it could be, is it a mosquito microbiome effect? Okay, so the microbiome, for anybody who doesn't know, the idea is an organism is not just an organism. An organism never lives in isolation. It's always in association with millions of different microbes. So it's entirely possible that a microbe within the saliva or the body of the mosquito might be causing this effect. So that's what they're testing. How do they test it? What's the actual treatment? Yes, so it's essentially plus minus antibiotics. Is there any other ones? Oh yeah, there's a couple others. What other things did they look at? Yeah, sex specific. Although I think that one's kind of obvious. Like male mosquitoes don't even bite. So obviously like their saliva is going to be like completely different. That's sort of obvious, but essentially like when they do that, they're kind of doing a technique where if you ever get to the point in science where you develop a measurable phenotype, you should milk that for everything it's worth and collect every piece of data that you can possibly collect from that. And it will make your publication more robust. So they kind of have this like sex specific thing, which is totally obvious, but they get data for it. So it's just like extra data. Yum, yum. Do they test that? I don't know. That might have been something I didn't hone in on. Yeah. Okay, so let's start. Oh, that's right. They do test, and I vaguely remember the effect was consistent. So like it's essentially like continuous production of whatever this thing is causing. Okay, so let's take a look at some of these data. First, let's try to grab this. Figure 2A and B. Okay, let's see if we can orient ourselves here. So first off, what's the treatment? The treatment is untreated or antibiotic. So this is the negative control, this is the test. Okay, and it's the same for this, same for this. So you'll see figures like this a lot. You don't need to get intimidated. Essentially, like this is the same experiment with something they're measuring here, something they're measuring here, something they're measuring here. So the question is, what is this? What is this? And what is this? Any thoughts? Yes. So these are some, I don't know much about these genes. Like these are just names of genes. CXCL2, I've never heard of it before. IL1B beta, but I don't study immunity. I'm assuming these are like immunity markers. So like if you get an infection, the first thing that you see that shoots up in response to infection is a certain quantity of expression of these genes. So what are they actually measuring on the y-axis? What is this? Presence of what? Copy number of what? No, look at the y-axis. This is not viral titer. Copy number of genes, or can you say it more? So I think you know you're correct, but you're not saying it correct. Why is copy number of genes wrong? It's not the genes, it's actually not the protein, it's in between the gene and the protein. Yes, it's the messenger RNA. So when I say expression, that's what they're measuring. They're measuring copy number. So again, this is the same thing. It's a QRT PCR, except in this case, they're not QRT PCRing for the virus. They're QRT PCRing for transcript of a specific gene, which is a marker of an immune response. So if you see this thing go up, it's like, oh shit, we're infected. There's something going on. And so what is the What's the conclusion from these data? But what's the conclusion? So saliva is, that's correct, is enhancing immune reaction, but only under what condition? So right. So simulate or synthesize that into one final conclusion. Saliva is enhancing immune reaction because of bacteria. Yes. It's not the cause. It's not the cause. So this is good. This is how science progresses. So remember, they have a list of hypotheses, and they do a first experiment on these microbiome hypothesis, and they get some preliminary data, which is this, which says maybe the microbiome is responsible. Okay. And then, but then here's where you get in trouble if you don't read all the figures. And this, I get in trouble for that a lot. Let's look at B. Now they're actually testing the same experiment, but look at the y-axis has changed. So what's the y-axis now? This is the copy number of the virus. So they're doing the same experiment, but now instead of measuring the immunity genes, they're measuring actual copies of the virus. And look what happens in this experiment. What happens in this experiment? It doesn't do anything. Like there might be a little effect, but it's not significant. So essentially, the microbiome has no effect on the virus phenotype. There is an effect on immunity, which makes like total sense. It's kind of obvious. Like why would the A up here, why was this kind of obvious? Like why would this be correct that you would see this, but you would not see this? Yeah, like the mosquito is biting and they have bacteria. So you're throwing bacteria into the bite site. So it's obvious like the organism is going to sense that it's being bit. It's being stimulated by some like bacteria. And so this makes sense. But the upregulation of these immunity genes in response to that microbiome is not causing the effect of the virus pathogenesis increase. Okay, so they are effectively able to rule out this hypothesis. Like there might be an interesting phenotype in that immunity thing, but it's not related to the thing that they're actually studying. Okay, so then let's get C and D. Let's see. So here, now what are they testing? Yeah, they're testing, is it a protein or not? So they have the saliva and heat-treated saliva. So in the heat-treated, you should be like denaturing the proteins. And what's the conclusion? Now you should get comfortable with reading this. What's the y-axis? It's number of virus. I don't know if you call it replicons or virions. It's the number of genomes of the virus, the RNA genomes. And what do they see? Yeah, there's a downward effect, a downward trend in essentially all the different tissues. So inoculation site, the skin, the spleen, and the serum. There's a downward trend, okay? If you are heat inactivating the saliva. So what is the synthesis conclusion from this? What would you conclude? Yeah, so the enhanced pathology is caused by a protein in the saliva. That would be sort of like the conclusion from these data. And then they test their obvious experiment, female versus male. And what's the conclusion here? What? Essentially, like the male saliva is not really doing anything. It's not, it's, if you look here, the male saliva, male saliva, male saliva, it's not really enhancing the copy number of the virus, but the female saliva is consistently. So again, that's kind of obvious, like males don't bite humans. So it's obvious that sort of like their saliva is going to be different. But if you're looking for whatever this thing is, don't bother looking in the males. It's in females. Okay. So these are some of their conclusions. So from here, they've kind of, okay, they figured out it is sex specific. Yes. It is a protein. Yes. Okay. Now what's the next obvious thing where you go? What protein? How are you going to figure that out? And this is pretty there's sort of like strategy, like you're trying to figure out some phenotype by some protein. It's pretty common what they did. What did they do? How do you answer that question? Did they?
I don't know.
Oh, that's an idea. That would be hard because most of the enzymes that break down proteins are kind of like cutting at like there are some specific ones, but most of the proteins are kind of like non-specific. So you'd sort of get like a general degradation. So I think that would not the best idea. Yeah, yeah. What did they do? Did you guys catch it? That came after. Mm-hmm. Can you explain that to everybody else, what you mean by that? That's correct. Well, even before you can blast, because blast requires that you have a known protein. So they can't use quote unquote blast to find the protein, but you're very close. What are they doing more precisely? Yeah, it's comparative genomics. Comparative genomics. Okay. And like, help me define that more specifically. Like how do they compare genomes and what genomes do they compare to get a good hint about what this thing could be? First, I think they even before, like you're implying that they know what protein it is, but even before they found what protein it was, they did comparative genomics. Which genomes would you want to compare? What? That's the type of genes. Which genomes would you want to compare? So actually, even before this, they tested the effect with anopheles. So they first did a test. One test was, do you see the same effect in anopheles spit? And what was the answer to that? The answer was no. I don't know what figure that was, and I'm not sure I grabbed it. But the answer was no. You don't see the effect in anopheles. So that now sets up the idea that you can compare, guess which genomes you can compare. Yeah, anopheles and Aedes. So you set up these two comparisons where anopheles is not going to have the factor and Aedes will have the factor. Okay. And then essentially, how did they screen for, actually don't know. Like through this comparison, they somehow got this sialokinin gene. But I don't actually know, like there's going to be a lot of differences between anopheles and Aedes genomes. So I don't know how they honed into that one specifically. Did anybody catch that if I missed it? That was definitely the next step. Anopheles mosquitoes lacks the ability. Are they citing a previous paper? I'm trying to find that on page 7. Other known ones. I see. Okay, so oh, that's right. They had that. So they probably, so I don't know exactly. I'd have to read it a few more times to know for sure. But they had this sort of setup where they know that it's going to, whatever it is, it's going to be different between between these two genomes. And then they probably developed kind of what you were saying, like a list of candidates that either are known proteins in the saliva, which people have perhaps studied and published before, or perhaps are known to, yeah, has some kind of like pre-characterized pattern. So they probably developed this list, and then they would check the list between the genomes to see like if there is any differences. And this one that was on their list was different, and so that became sort of like their top candidate. And then the final experiment, then what's the final experiment that you do? Yes. So then the final like wrap up, like prove it to me. You make the protein alone, and instead of saliva, you inject the protein. Okay, let me see if I can get the data for that. I think that is I'm looking for it. Might take me a second. I'm seeing a B, C. Somebody help me find that experiment. Oh, here it is. Yeah. Let's grab this. And here's where they're aligning this protein and finding its differences. And then here's the data. So here's again, like, here's like one of this paper has a lot of like solid figures where it's like, bam, here's the cool phenotype. And so here is, let's look at, what is this y-axis? Here's copy numbers of the virus. This is the key thing. Copy numbers of the virus alone, virus plus a specific protein, and it goes up. Virus alone, virus plus this protein, and it goes up. So they've shown that they can reconstitute the phenotype with this specific individual protein. And that's kind of like the end take home, right? Okay, so let's do some wrap up. Like, what would they do from here? What would be next? What? A what? What did you say? Human trial of what? Like, what you want to inject it to people to make their viruses worse?
That perhaps can block the protein and then decrease the viral infection. However, last thought that they said is like, what is going to happen if we have a hypersensitivity reaction?
Yeah.
Like this component, those people see that be like people now are gonna have allergic reactions to proteins that have vaccines.
Yeah. Yeah, I think you hit on it, and perhaps essentially like you've identified a target, right? Like perhaps you could inhibit this interaction somehow, and maybe you would make it less likely for people to get some particular virus. You could, although I kind of feel like so that you definitely could, like, you could try to modify it, a mosquito. But my thought is like, if you're going to modify the mosquito, you could do infinite possibilities. Why is this one the one you want to do? You know what I mean? My thought is there's better modifications out there, although you could do that. Also, the next thing that I would want to see, just to be like a little bit more rigorous, you'd like to see like a knockout. Like they've tested it with the protein, but you can make knockouts of mosquitoes. And so they could make a mosquito knockout that doesn't have the sialokinin gene, then do the same biting experiments and see if it works. Or maybe they just wanted to like wrap it up. Yeah.
How would that be different than using the broccoli?
It's a tighter, it's a similar experiment, yes, where they're doing anopheles saliva, which, oh, I see what you're saying. it's like the reciprocal of that. It's a cleaner experiment because one thought is sialokinin, one criticism is sialokinin might be one component contributing to this phenotype. And if they're kind of concluding perhaps it's the only one, like it's the main cause. And if it is the main cause, then knock it out in the 80s and then you will exactly replicate the phenotype. But if you knock it out and you get like half the phenotype, that shows you that there's something else. You know what I mean? So it's sort of like it's a tighter experiment than the anopheles. Although I agree with you that the anopheles is a good.
Figure right above,
Figure right above
there,
this thing.
yeah, D and B right there.
Yeah. Yes. And then it reconstitutes. Yeah.
Yeah, I think you mean.
No, this is a good experiment, and this suggests this suggests like it is sufficient. It's just another thing you could do. I don't know if I would actually do the knockout. You bring up a good point. Yeah. Yes, you could do it for more viruses. Mm-hmm.
That's on the prison, he not transmitted by all.
Wait, well, they're using anytime they're using the anopheles, that's sort of like a negative control. Yeah, where they don't have the sialokinin, unless they're adding it.
They already transmit
Anophely? No, they only transmit that young. I don't know. Yeah, I think it's young. I think. Yeah. Anyway. Any final questions? All right, good. Very good. Okay, so we'll do, let me stop. We will do